Hello dear colleagues, this is our sterilization room and I'll show you some of the machinery we use for sterilization and disinfection. Now, sterilization is a method that would kill all the microorganisms on a specific surface or in a specific substance, including the bacterial spores, which are the most resistant forms of life. Now, this machine is the hot air oven or hot air sterilizer. So here you could see there are two thermometers. One of them shows the temperature that is outside, the other one shows the temperature that is inside. Inside, we usually maintain a temperature of about 160-180 Celsius degrees. So here we sterilize all the glass objects in a microbiology lab. Those flasks, those petri dishes, tubes, pipettes, and so on. So what do we need to do first? First we need a small indicator paper, which has this light blue color. It needs to be placed among all those objects. But of course, the objects are not sterilized immediately. The glass objects first need to be soaked for 24 hours in some disinfective solution. Then, again for 24 hours, they need to be cleaned, they need to be rinsed in several buckets of distilled water. After that, all the glass objects need to be completely dry before we place them into the hot air oven. Because if we place wet glass on such high temperature, there is a chance the glass may break. The reason is water has a much different heat release quotient than glass, so it is quite a possible thing. Our aim is not just to have a sterile glass, but that glass to be in the shape of tubes, flasks, pipettes, and so on. So, here we close this metallic cabinet. Then we turn it into the wiring, and once the temperature reaches the designated degrees, let's say 160 degrees, we need to keep it for at least 2 hours. Or if it is 180 degrees, that should be maintained for at least 1 hour. So after this treatment, spores should be killed. The point is, we should never over-trust the machine. For that reason was, in fact, the indicator paper that I placed inside. What is the reason for this? Take a look here. This is our logbook. In the logbook, we write when did the sterilization process start, when did it end, and do you see the difference? The way the paper changed its color. It turned into a deeper blue. That happens once the temperature reaches 160 degrees. So after that, the person who holds the sterilization process needs to sign and needs to stick the indicator paper inside. So this is a proof that everything was okay and the designated temperature was really reached and maintained. Okay, now take a look here. This machine, which has the shape of a rocket, it's in fact not a rocket. This is a giant kettle, or scientifically said, it's the Koch device, Koch sterilizer or Koch apparatus. As you may see, there is some plate which has holes inside. In the lower part there is water. All the objects for sterilization will be placed here inside. It is in fact still hot, it has worked today. Well, it's not hot, it's warm. So we place, for example, all the jars with agars or other nutritive media inside. We place the lid. Probably you see there is a hole on the lid. So when we turn on into the wiring, the water temperature starts to increase. Once it reaches the point of boiling, which is about 100 Celsius degrees, you will see steam coming up. So spores cannot be killed by 100 degrees. So just boiling or just flowing steam is not enough to kill the spores. However, in some cases, we cannot lift the temperature above 100 degrees because nutritive media will lose their nutrient properties. Carbohydrates will turn to caramel, proteins will denaturate. So we cannot afford this. Then we have a paradox. How will we kill the spores if we cannot increase the temperature? Well, the answer is the so-called fraction sterilization. Once we reach 30 minutes with this procedure, we turn the machine off, wait for the steam to cool, and then take the nutritive media out. After that, we will place them in a thermostat at 37 Celsius degrees, where there would be perfect conditions for the spores to turn back to vegetative cells for 24 hours. On the next day, we will repeat the procedure for another 30 minutes inside the Koch sterilizer. And in order to be sure there are some lazy spores left, we will repeat this once again. So 3 consecutive days at 100 degrees, and the spores will be cheated, figuratively, to turn to vegetative cells, which we will kill. So this is the way we achieve sterile nutritive media. Now, take a look here. Those 2 machines, which look like some laundry machines, are not laundry machines. These are the autoclaves. Or, in fact, they are some giant cooking pots under pressure. Well, of course, they are not for cooking, they are for sterilization. All the objects are placed into a bucket. You see, this is all prepared to be sterilized on the next morning. This is all with some used tubes, agars, gelatins, broths, and so on. So these microorganisms need to be killed. The principle is just the same. On the lower portion there will be water. Then, when the temperature increases, steam will go up. However, you could see there is a valve on the top. There is no opening like in the Koch sterilizer. It is hermetically sealed. So the steam has no way out. Or at least it has no way out until the steam reaches a specific increased pressure. So here we can put the pressure and the temperature we need. If we increase the pressure of steam by half an atmosphere, then the boiling temperature of water will increase to 121 degrees. If it's 1 additional atmosphere of pressure, then boiling temperature of water increases to even over 130 degrees. Well, such temperatures of 120 or more are enough to kill the spores. So here the pressure serves only to elevate the boiling temperature of water. Like 10 or 15 minutes in the autoclave will be sufficient to kill all of the bacterial spores. This looks like a paradox. Take a look again. The hot air oven needs more than 1 hour for temperature at least 160 degrees, while the steam under pressure autoclaving needs lower temperature and much less time. The answer to this paradox is steam and water-containing environment. Hot air is an insulator, so it takes much more time for the spores to uptake the heat. On the other hand, water is also a very good environment. Steam is a good environment for the hydrogen bonds which make the tertiary structure of biomolecules to form. Yet, in the presence of water and steam, hydrogen bonds are much more easily disrupted too. So the biomolecules need much less time to be destroyed. Well, the most resistant biomolecule is DNA. So DNA sometimes survives even after the spores are dead. And that means that if we want to sterilize some objects, let's say some regions, to perform PCR techniques or some other methods that we will deal with DNA, we need a longer procedure which takes about 20, not 10 or 15, but 20 minutes of autoclaving. That should be specifically designed when you leave the objects for sterilization. Why do we have two autoclaves? Because one of them is figuratively clean and the other one is figuratively dirty. The clean autoclave, of course, it contains a lot of microorganisms, but we put there objects which we don't consider to be really contaminated with pathogenic infectious material. While the dirty autoclave, here we will sterilize only objects which we know per se that have been inoculated, that have been contaminated with really infectious material. And we should not mix the objects between two autoclaves. Of course, okay, we need one more machine. That old Russian machine has no electronics, pure hydraulics. This is the third autoclave. In case one of the other two goes out of order, we should always have a replacement. So these are some of the most important machines. This is, by the way, a water distiller. Of course, everything in a microbiology lab should be prepared with the distilled water. This machine here is a novel generation. It is not just a water distiller, it is water deionizer for chemically clear water. So it is UV disinfected, it has also reverse osmosis and also distiller. Also, in this machine, this is not for sterilization, but there is an ice machine. When we perform genetic experiments like PCR, for example, all the reagents should be kept and stored in ice. Otherwise, the activity of all those reagents will soon be over and expired. So, this is end of part 1. See you in a minute.